In spy movies, an undercover agent is often given a task to find their target and eliminate a threat, all before the world is destroyed. These agents need to be trustworthy, use stealth, and are often given cool gadgets that help them to, in the end, save the world from catastrophe. Unfortunately, there are, however, many lives that are taken by a growing threat that no gadget in these agents' arsenal can combat. This is because the threat lives at the microscopic level and is launched by antibiotic-resistant bacteria. In the past, these bacterial infections could be eliminated by a cool group of gadgets called antibiotics, and some bacteria have evolved to become resistant to these drugs. Unlike other bad guys, no agent can physically apprehend these bacteria, so my research is focused on enlisting the help of a new type of agent, the bacteriophage, aka Agent 5. Bacteriophages are viruses that naturally infect very specific bacteria through the use of a gadget called receptor-binding proteins. These are specialized proteins located at the tips of their leg-like structures that scan the bacterial surface for their target. Once found, they irreversibly anchor the bacteriophage to the surface of the cell, triggering DNA injection, the start of the infection process. The cell is quickly rewired to produce more bacteriophage agents, which rupture out of the cell and find new targets. This type of bacteriophage would be the perfect agent, but just like some spies, not all bacteriophages can be trusted. Some bacteriophages carry toxins or even antibiotic resistance genes that can be transferred to bacterial cells if the bacteriophage DNA does not replicate but incorporates itself into the bacterial chromosome. If used, these bacteriophages would betray us by joining forces with the bacteria and actually contribute towards antibiotic resistance and pathogenicity. Consequently, understanding the characteristics of each bacteriophage is crucial in selecting the perfect agent. My research focuses on further understanding the initial step of the infection process, target recognition, and DNA injection. Further understanding how these receptor-binding proteins, or scanning gadgets, work could potentially allow us to adapt the scanners to only target antibiotic-resistant bacteria, and not the healthy bacteria found in our gut. Due to the specificity of these receptor-binding proteins, the bacteriophage will not harm human cells, because they only target proteins that are found on the bacterial cell's surface. Insights into how receptor-binding proteins work could allow us to enlist the help of bacteriophage agents and their scanning gadgets to combat the growing threat of antibiotic-resistant infections.